Today we're talking with Lindsay Cameron, who's currently postdocing at Stanford with Rob Malenka and Carl Dazaroth, which is pretty exciting, I'm sure. And you've been doing a lot of amazing work in psychedelic mechanisms and psychedelic science prior to that in David Olson's lab at UC Davis, which is where I think I might have first met you. Thanks for jumping on for a chat today.
Of course, it's so much fun to be here.
Yeah. Yeah, so I guess one of the things I'm always curious about with people, if we've been diving deep into the sort of mechanisms around serotonergic psychedelics, is, how did you get interested in this particular field and this line of research?
Yeah, so I was just a brand new neuroscience student at UC Davis, and I actually did my undergrad in pharmacology, like drug design and development, and with a heavy background in organic chemistry. And I was totally going to switch fields. I actually felt like pharma companies were evil and they were just profiting off of sick people. And I was very morally against this. And I was transitioning to study stem cells, actually, and stem cell transplantation. And so I rotated through Dr. Olson's lab just to kind of check out and see what it was about. And it was awesome. It was so cool. He had just opened his lab, so it was brand new. And like three things were up and running. And I loved David's charisma and passion. And it seemed like they had some really cool projects going on. At first, I kind of didn't take it that seriously. I just thought it was kind of like a fun thing to do. And then the lab that I wanted to join, which was a stem cell lab, actually their funding fell through right when I wanted to
Right.
join, and it wasn't quite as great a fit as I thought it was going to be. So I decided to join the Olson Lab, and I was really excited because at this point, it was 2016. Basically, no neuro research was done on psychedelics, but there were like a couple of those Roland Griffiths papers that were out
Mm-hmm.
that showed that it helped with depression and anxiety in end-of-term cancer patients. And I was like, that's really cool. But the basically the mechanism behind how it worked was totally not known. Um, and this did seem to be a therapy that was more curative than actually just treating the symptoms, which was my whole hang up on the pharma industry. And I was like, my last hang up, my last hurdle before joining the Olson Lab was if I study psychedelics, no one will ever take me seriously as a scientist. They're going to think I'm a crazy hippie, and no one will ever take me seriously. And I was weighing all these things, and honestly, it was the cool science and David's charisma. And he was so smart and fun as a PI. And I was like, screw it. I'm going to do it. I'm going to study psychedelics, and then we'll deal with the consequences later. And so that kind of launched me into studying psychedelics for the first time and looking at like what they actually did to the brain to achieve these therapeutic effects in people. And a couple years into my PhD, How to Change Your Mind came out. And I
Right.
think that
Yeah.
was really the pivotal point where all of a sudden everybody became interested in psychedelics. And so it turned out kind of great for me, but I totally didn't know that when I started my PhD.
Yeah, I think you're actually getting into it at a pretty interesting time, like kind of even a few years. I think David's lab was interested in this area probably a few years before the sort of the more mainstream hype cycle that's taken over.
Totally.
I don't know if you've ever spoken to David Nichols or Chuck Nichols about David's advice to Chuck, but he basically said to him, don't study psychedelics. It's an absolute death knell for your career. And he's one of a handful of people, along with sort of Brian Roth and others, who've managed to successfully have their labs funded studying these super interesting compounds for the past few decades. Whereas, most people probably did treat it as a bit of fringe science, even at the time that you're talking about. So yeah, it was
Totally.
pretty exciting
It
to see all
was
this coming out of David's lab.
Yeah, it was super, super fun. And one thing that was really cool was that these drugs, like they work in humans already, right? Which is not like a lot of normal drug discovery.
Mm-hmm.
So, and yeah, they have incredibly powerful effects on the brain, but no one knew how they worked. It was just this
Mm-hmm.
black hole. You could study whatever you wanted, and it was brand
Yeah,
new
yeah.
data for the field. Like everything was publishable, and you had no competition because when I started, there were only like three labs in the whole country with a Schedule One license or something.
Mm-hmm.
So there was like no competition. All data was new data. It was this glorious way to enter science and just see what happens when you give drugs to cells or mice.
Yeah, this sort of bedside to bench reverse translation, which is definitely not that common in drug development, is what I feel the field is moving in terms of better understanding these mechanisms and what might be going on. So I remember a few years ago, there's a really interesting set of articles. Like I think it was David Yaden on one of them as an author and David Olson on the other. And it was essentially the titles of each of the articles in ACS Pharmacology and Translational Science were the subjective effects of psychedelic drugs are essential for their therapeutic activity. And the other one from David Olson was the subjective effects of psychedelics are not essential for their therapeutic activity. And these are like bookended in the exact same issue of ACS Pharmacology and Translational Science, which I thought was amusing. I sort of have my own views on mood disorders versus other areas of neuropsychiatry, maybe on neurology. But what's your take on how critical the sort of subjective effects of psychedelics are for their therapeutic activities?
Yeah, so I do not think the subjective effects are necessary, but they might help. So having these kind of profound experiences can definitely help people or solidify or drive home a point that people need to do, especially when paired with psychotherapy. But I don't necessarily think that they are necessary. I think the putting your brain into some sort of plastic state is kind of what really helps. And I don't think that you need the subjective effects to get there. They're indicated for many different populations, including those with schizophrenia or those with family members that have schizophrenia. A lot of them are now screening out people with any form of cardiovascular issues. They're being screened out of trials. So offering compounds that may be able to still help these populations that need help, but aren't contraindicated is also great. I also want to say that I think therapy will help either way. So I think therapy in conjunction with a psychedelic can really help. And I think that therapy in conjunction with a non-hallucinogenic analog, or sometimes even just therapy alone, can help. So I think that like having therapy generally accelerates and putting your mind into this plastic state can help. And I think that, yeah, it's possible that the psychedelic effect may drive home the point more strongly or maybe make the effect more long lasting. Those things have not been studied. It's possible that that's an option there. I do think that with some patients, some populations don't really need this insane self-exploration. They kind of know what they need to do, but they need a push in the right direction or some motivation to kind of get there. And I think that those are often people where SSRIs might work. Um, and I think that this is a way to get them there in a fast-acting way
Mm-hmm.
that may help
Yeah.
without all these crazy subjective effects.
Yeah, I think that's a probably a sensible position. This idea that a lot of people view SSRIs, they have this sort of latency to onset, but part of what they do for a lot of people is sort of enable them to create change in their own lives, especially people with pretty serious mood disorders. So the idea that you might be able to do this more rapidly with some of these mechanisms is certainly pretty interesting. I'd say yet to be fully clinically validated, but very
Mhm.
exciting. So just on that point, I guess I've spoken to quite a few people about areas outside mood disorders, but particularly in sort of areas of chronic pain, phantom limb pain, or cluster headaches in neurology. How critical do you think the sort of 5-HT2A mediated mechanisms are across disorders versus maybe some of the other polypharmacological actions that many psychedelics have?
For sure. So I don't want to talk about chronic pain too much just
Yeah.
because I know some super experts in that field
Hmm.
that I am happy to point you to if you want to talk to. I think that so there's a lot of different components to pain. Um, Part of which is actually physically feeling pain, some of which is depression or anxiety or mental health associated with that. And the idea is that if you can kind of strengthen your mental health or strengthen your top-down control, you can kind of have more control over perception or
Mm.
um, yeah, just control over what you're able to do without the pain getting in the way so that there's more cognitive kind of influence. And so the question then becomes, is it actually reducing the pain or is it just increasing your cognitive control over the pain?
Yeah, very important distinction for certain forms of chronic pain, right? Where it's not so much that you're actually treating the pain itself, but your ability to deal with it.
Mhm.
Yeah.
And one thing that I think Tess is doing that I think is very cool is she's looking at people's relationship with pain and how that changes before and after. So that's also kind of cool. There's all these kind of different aspects that you can deal with it. I suspect that it doesn't change the sensation of pain directly. but rather it would change your cognitive control over it.
I think there's more sort of data supporting things like cluster headache, where there's some sort of community-driven efforts. You can get on Reddit and find a lot of people who are treating cluster headaches with their
Mhm.
own serotonergic psychedelics in various ways, and 2-broma LSD has a big history there. But yeah, pain's sort of one of these interesting and emerging areas where a lot of the mechanism's not understood.
I think that actually Boris Heifetz and Josh Woolley are just finishing up a trial on psilocybin for chronic low back pain.
Right, very
So
cool.
they will actually have some more very interesting data on pain that is actually through a clinical trial now, which is
Yeah.
kind of cool.
Yeah, very cool. I think you may have seen some of these papers that have attempted to look at the mechanisms, preclinical mechanisms for psilocybin in various rodent pain models. And there's sort of
Mhm.
a bit of a challenge to reproduce data across a lot of those models, which speaks a lot to the blind spots we have in terms of mechanistic work here.
Totally.
Yeah, which brings me to a paper which I think you're an author on, a preprint that I read a little while ago from Rob Malenka's lab with this sort of SciBank project, which is this pretty interesting idea, I think, to try and have a number of different labs in California, all using the exact same methods in a number of different sort of preclinical translational models and trying to replicate the basic effects of psilocybin at several doses in these very widely used models. So could you give us a bit of a summary of sort of that preprint and what was found? Because I thought it was pretty interesting and definitely the sort of work that needs to be done in this area.
Yeah. So I guess the overall view for anyone that hasn't heard of this paper, it's basically trying to replicate the effects of psilocybin across multiple different places. So there's two labs at Berkeley, two labs at UCSF, and two labs at Stanford. And everyone basically tries to run the same protocol and then compare to see what they get. We want to see how replicable it is to get the same results that everyone's been reporting in the field. And the answer is it's sometimes very hard. So,
Yeah.
some things that are very replicable are animals get very anxious immediately after you give them drug. That seems very replicable. That seems very fair. The antidepressant effects seem to be a little hit or miss, which is interesting. And one reason why I really like this paper is because I think generally as scientists, someone will publish a paper and it will say psilocybin works in the forced swim test or tails it. Psilocybin works for depression in animal models, you know? And then anyone who tries to run this themselves and can't get it to work, they assume that they're doing it wrong because they can't
Yeah.
replicate what this other person did. And what I like about this paper is it's saying, stop gaslighting yourself. Sometimes it doesn't replicate. And even across these six different labs, we get hit or miss on if it works or not.
Hmm.
So, what I like about it is for our community, for our researchers here, to kind of stop gaslighting themselves. And sometimes it works and sometimes it doesn't.
Hmm.
And I think that this is useful for multiple different reasons. One, so that we can have more clear data. Using animal models is very useful because they don't have a placebo effect the way that humans do. So when you poke a mouse with a needle and give it psilocybin and it gets high, the mouse doesn't know if it's going to feel better or not
Yeah.
afterwards. Versus if you do this with a human, it's very easy to tell if you got psilocybin or saline. And then just by undergoing an experience, you're going to be functionally unblinded. You're going to know which one you got, and therefore you can have these strong expectations. So it's very hard to run these placebo trials in humans. Um, yes. One thing I do wish the SciBank paper did was test different doses and do an entire
Mm.
dose response. Um, this was a area that I argued that I got vetoed out of, but I would have gone much higher on the doses of psilocybin than that
Mm.
were used
What,
in
what
that
what other,
study.
at least two doses were used, right? If I remember correctly, something like this. It was low, moderate dose and something a little bit higher, but both sort of doses that can be found in the literature elsewhere. Is that
Yeah.
right? Yeah.
So I am definitely a fan of higher doses, and my argument is that it would probably be more replicable if you used a higher dose. And so, yeah, so one of the reviewer comments that we got was that we should add some more doses in there. And I fully agree with that. You know, it's
Hmm.
better to give someone like the doses that are being used in clinical trials are often higher doses are what we're seeing the effects in than like a little museum
Mm.
dose where you see some cool colors on the
Hmm.
wall, but it's not a
Yeah.
full-blown experience.
Yeah.
Um, Yeah, so there's a bunch of stuff to unpack with that SciBank paper.
Mm.
Um, but I think it's all good. I think it's good for the field.
Yeah, absolutely.
Check. And
Yeah.
we're seeing this with clinical trials too, where sometimes it doesn't work. And that hype of psychedelics will cure everything is finally bursting, I think, which I think is a good thing. I think we need to be realistic about these medicines. And so I think it'll be good and reflective of
Mm.
like clinical data and stuff coming out now.
Yeah, yeah. I'm very skeptical of any claim that a single drug is a panacea for everything that ails us. So that triggers my inherent sort of skepticism on a lot of fronts. But I remember
$100%.
talking to Rob about the SciBank paper, and he laughed it off that it's not especially high impact because it's a lot of sort of negative results. And we've discussed this idea that that's actually extremely important, especially in translational neuroscience, where things are published once in a big journal and often they're not replicated, but there's no sort of incentive for scientists to actually publish the level of reproducibility of those experiments in terms of actually publishing the experiments that work and those that don't work and are harder to replicate. So this is sort of
Mhm.
a larger issue in sort of preclinical or translational neuroscience at the moment, I would say.
Yeah.
Yeah, yeah, very cool. So what are you working on at the moment? What's taking up a lot of your time and what are you excited about?
I am working on way too many things. But I love them all. And so every time I make a discovery, I end up with a new project. So that's where I'm at. So I went down this rabbit hole, actually. I came to Stanford to study psilocybin for treating opioid use disorder. And I was really interested in studying more of addiction and substance use disorder rather than depression and anxiety because it's just an easier way to translate to humans. If an animal takes more drugs, that means they like it. And that's pretty similar
Yeah.
to humans.
There's slightly more objective measures than we think about in other areas of psychiatry and in rodents for sure.
Yeah. So that was very nice. It was kind of how I finished off my PhD with tabernanthalog, was looking at heroin self-administration, and that was very cool. And I have pivoted to the field of looking at addiction and addiction drugs and looking at psilocybin for treating specifically fentanyl use disorder. Which made me go down a rabbit hole and I accidentally made some discoveries about fentanyl, which is kind of cool. So now I have multiple projects that are all on opioids and not about psychedelics at all. Uh, and I'm trying to wrap them all up and publish them in the near future.
Yeah, that can be the challenge of recalling my sort of postdoc days at Stanford. There's huge amounts of intellect there and a lot of clever people with a lot of interesting ideas and maybe too many ideas to pursue simultaneously. So you need to sort of rein it in, have some focus at some point as well. But yeah, very,
Yeah.
very exciting stuff.
Yeah, it's very fun. Glad to be here. I should finish my postdoc at some point.
Awesome.
Hopefully in the next year or two we'll get there.
Yep. And what do you think about doing next?
I'd love to apply for some faculty positions.
Um,
If anyone's hiring, I don't know what's going on with the government these days and if universities have decided to put out calls, but if they are, I'd like to go on the job market sometime soon. I just got my K-99, so I'm very
oh,
excited.
congrats.
Thank you.
Yeah, that's
So
exciting.
hopefully that'll help me if when people are hiring.
Awesome. Yeah, no, I'm sure it will. Alrighty. Well, thanks so much for your time, Lindsay. It was yeah, really great chatting and best of luck with all the experiments and wrapping up the postdoc.
Thank you.
Alrighty. See you soon.
See you.